Hi everyone, it's Mr. Sinti, and I'd like to talk to you today about primary growth. And primary growth is going to be a conversation that we're going to have that's going to sort of circle around the idea of apical meristems, which are, if you recall from a previous discussion, those sort of well springs of cells that are capable of mitotic division even as a plant gets older and older and older. And so primary growth is going to be about the plant growing up towards the sky and down towards the center of the earth, or up and down. But what's interesting is this primary growth is going to also lend us to a conversation about the different tissues, ground, vascular, and dermal, and it sort of is going to wrap up a conversation that we've had before about sort of differences between monocots and dicots. So all of that is going to happen in today's conversation. And so without further ado, let's just attack that. And so when we're talking about primary growth, we're talking about the roots and the shoots, because this is where the apical meristems are, the terminal buds that produce up and down. And so let's just sort of jump into that and say, for example, when we look into that, let's see, let's go right into that. Whoops, let's see, sorry about that. Let's go here, and then let's go there and there. And so here we are at the apical meristem of a root, or the terminal apex of a root. And so let me draw your attention to this region right in here. Do you see this apical meristem? If you were to go even higher under the microscope, you'll be able to see these cells undergoing mitosis. This is a good place to look. Sort of longitudinal section of a root. You can see the cells dividing. Look, if you look really closely, you can see a cell undergoing cytokinesis right there. And so right here is where the cells are dividing. But do you notice here on the very tip of the root, there's something called the root cap. And the root cap are cells that are no longer dividing. Not only are they not dividing, but they're dead, and they provide sort of like a thimble, like to the root itself, because under the soil, soil can be kind of scratchy, and it's abrasive, and so it'll tear away at the root, and those cells are sloughed off without it damaging the cells here in the meristem that are actively dividing. So the root cap plays a protective role. It doesn't divide itself, but yet it is the consequence of these cells dividing right in here. And these cells actually secrete a little bit of a slime which helps to lubricate the root as it moves through the soil. So growth, what we're talking about is it occurs near the tip of the root. And so look down here. Do you see here's the root cap, and this sort of circular region right in here is the apical meristem. Do you see that? Apical meristem. These are the cells that are dividing right in here. There's a region in between the apical meristem and the root cap itself. This grouping of cells right in here is called the quiescent center. That's kind of a cool word. It's not restrictive to biology. It just means kind of a quiet area, or a reserved individual is sometimes referred to as quiescent. So it's a bunch of cells right in this area that are sort of resistant to, they're not dividing as quickly as the apical meristem, and they're sort of resistant to trouble, and so they can become meristematic if called into it. And so what I want to say about this apical meristem right in here is that the apical meristem can be further divided into three kinds of primary meristems. So this is a primary meristem, meaning that it involves growth. And when the cells are dividing, the cells are going in an upward direction as they mature. And so this region right in here is the zone of cell division, right in here. So this is mitosis. And this region can be further divided into three kinds. And let's go right in here. Do you see this one that says procambium? Procambium are the cells that are dividing that will become, as they elongate, in other words, they get larger in size and then specialize, this will become vascular tissue. So procambium matures into vascular tissue. And then once it's vascular tissue, it's no longer dividing. So notice the vascular tissues here in purple. That cylinder, which is found in the root, is called the stele. Right in here, the stele. Sorry for the terms, but that's the stele. This yellow tissue right in here is ground tissue. So it's vascular, ground, dermal. Dermal's blue. Ground is yellow. So what becomes, when the cells divide, what will become the ground tissue? Well, it's called ground meristem. Yay, you can remember that. Ground meristem will become ground tissue. So as the cells elongate and move up, they will specialize into this ground tissue. Now, what is this ground tissue called right here between the stele and the epidermis? This is called the cortex. If you recall, the function of the cortex in the root is that of storage. It has those leucoplasts in there. It stores starch. And so that's the function of the cortex. And so the third primary meristem, right over here, shown in blue, is called the protoderm. In other words, it will become the epidermis. So as the cells divide, and then they get a little bit larger and longer in length, notice here they become the dermal tissue. The epidermis can be specialized into root hairs. Look at this, maximizing surface area for better reabsorption. So there's three zones. There's kind of a, they're not so distinct. The truth is there's a little overlap between the zone of elongation as the cells get longer and as they mature. Notice how it's a little overlap. So you got three zones. Let me sort of emphasize this with a pen. You got zone right here of cell division, and then the cells get a little bit larger, and then they mature and differentiate. And what are they differentiating into? Into the three tissues: the epidermis, the cortex, and the stele. In other words, dermal, ground, and vascular, right there. And so there's three primary meristems in the root. I hope that was clear. And so here's again a close-up of the root. Here's the root cap. Here's the apical meristem, where the three primary meristems are: the protoderm, the procambium, and the vascular cambium as well. And so those will divide through mitosis, and this region right in here is called the quiescent center. And so this quiescent center I mentioned before is sort of resistant to damage, and so those cells can sort of are like bench players in a sport. They can come in, or bullpen. They can come in as the starter pitcher becomes damaged, if you will. And so these primary meristems, protoderm, procambium, and ground meristem, will eventually become the three tissue types: the blue, the yellow, and the purple, right there. And so here they are, the blue, the yellow, and the purple. So protoderm, ground meristem, and procambium, right there. And so another, as a cold drawing, here's the root cap right here. Here's the apical meristem, the cells that are undergoing mitosis. Here's the quiescent center. Notice here, look at this. As the cells move upward, they're no longer dividing. So this is where they elongate. They can get really, really big. If you take a look at this under the microscope, these cells are huge. And you can see here in the center is the vascular tissue, or stele, and then on either side is the cortex. If you think of it like this is a root, I don't know if you're getting a full sense of this, but if you make it three-dimensional, this stele is a circle in the center right in here. If you recall that, like in a carrot, there's like this central vascular tissue, and then that's the stele, and then on the outer parts is the cortex, and that's where the starch is stored. Here's the epidermis with the root hairs. Look here, here's the piths in the xylem, which will allow water moving between the cells. So this zone of maturation is when the cells begin to specialize into the different tissues, either dermal, ground, or vascular. And so what we have, again, just to sort of really drive this point home, is these primary meristems in the root give rise to the epidermis, which is right here, this outer layer of cells, this inner layer called the cortex, and then this vascular cylinder called the stele, right there. And what's interesting about this is here's a close-up of it's a corner of the stele. This is a big look at the cortex. It's purple because the cells are staining, because they're containing starch, and then here's the dermal tissue right in here. Well, I have some better pictures of the stele coming up, but again, here's the The protoderm will eventually give rise to the epidermis, which will eventually elongate and then specialized into here's root cells. And why is that? It greatly increases surface area and it absorbs minerals, especially water. Here's a really nice shot of the cortex right here in purple, which is ground tissue, and then the stele, which is the vascular bundle inside the root. The tissue that you see here in red that sort of looks like a Y shaped is the xylem. This greenish tissue over here is stained, which is the phloem. And then this outer ring is called the endoderm, as opposed to epiderm. Epi is on top of or on the outside. Endoderm makes up the outer portion of the stele. There's even a cell layer right below the endoderm called the pericycle. I'll come back to that a little bit later. And so this procambium, which is a primary meristem in the apical meristem of the root, will eventually give rise to the stele, which is composed of vascular tissue, xylem, and phloem. Here's a cross, sort of a cross of the xylem. Look how thick those cells are of the vessel elements, and those are because they have a secondary cell wall. It sort of forms like cells radiating like this, and then the phloem cells are right over there. Notice here, do you see here in the endoderm? Do you notice how these cells are a little red right in here? They're red because they're producing this wax that surrounds them, called the Casparian strip. I'll come back to this later. It's really cool. It prevents fluid from passing into the stele that is not wanted. In other words, there could be some toxics in the fluid that's moving in between plant cells, and you don't want that to get into the stele, because this is sort of the elevator of the plant, if you think about it. And so I like to think of it as like you're entering a building. First thing is it's hard to get in to the building because of security. For example, number one is the epidermis, and so it's not going to let anything in. And then if you do get past that, you can move through number two, which is the cortex or ground tissue. You can sort of go in between cells. If you travel through a cell, you'll have to go through the membrane, and of course the cell membrane is selective, so if you're unwanted, you won't be able to get in. But you can pass on the outside of cells. But you're not going to pass through the number three, which is the stele. That inner layer right in here called the endodermis has that Casparian strip which prevents that. And so this cross is the xylem, and then over here is the phloem. So vascular tissue, ground tissue, dermal tissue. This is the root, a dicot root. And the dicot root is known for this sort of cross shape. Here's the endodermis, the pericycle, which is this one cell layer underneath. Here's the xylem, here's the phloem. Here's the cortex, those little, sort of looks like it has the measles, which are little leucoplasts of starch. Found this picture. This one's really cool too. You can see here, this is a nice shot of the stele. Notice here you have your xylem here, and then just on the outside of that is this pericycle, and then just on the outside of that is the endoderm in purple, endodermis, right there. That's kind of neat. Here's the root cap, apical meristem. Real close-up of this. Here's the endoderm. Let me highlight it. Here's the endoderm cells right here that go around. One could say that the endoderm is considered technically the innermost layer of the cortex. And then you have this pericycle. And it's like, I mentioned that before, what's the story? It's this cell layer just on the inside of the endoderm. And why it's significant, I'll just mention it now, is that these cells are capable of division. And if they're capable of division, what can happen is it can bust through the endoderm, and it can actually form a lateral root. When I say lateral root is when you have like branches coming like this off of roots. It's like, how do those branches form? Well, they form when the pericycle becomes meristematic, and it starts to penetrate through the endoderm and through the cortex. And so Here's endoderm, pericycle, phloem, and xylem. Notice how thick the xylem is because of the secondary cell wall. By the way, what cell type is this? It's sclerenchyma, secondary cell wall. So this is I like this picture a lot. This is a sort of comparing and contrasting what a dicot root looks like. Dicot root is what we've been looking at primarily. We've been looking at the xylem, which is across here. Here's the phloem, which is in between. Here's that pericycle, I was just talking about it, and then the endoderm, and then the cortex, and then the epidermis. And you're like, yes. But then in the monocot, like for example in grass, for example, fibrous roots of grass, what you're going to see is it's not going to be a cross in the center of the stele like that, but instead the xylem are going to be in these little bundles right in here, and the phloem is in between the bundles. And then you have this little region in the center called pith, which gives support. It's a type of ground tissue, parenchyma cells. Pith, which provides some support for the root. And then here it has cortex and epidermis. Let's take a look at the real monocot root. So here's the real monocot root. Dermal tissue, ground tissue, vascular tissue. And then notice how the big circles right here are the xylem. All these little cells in between are the phloem, and then has this pith area. Here's a picture of a branch emerging. Again, monocot root. Xylem cells are a little thicker. You can see here, phloem is all in between. Right in here, here's the cortex towards the outside. The pith would be on the inside. Here's a real close-up of the endoderm right in here. Here's the pericycle, the cortex over on this side. If I were to ask you, like, what is that? You'd say, oh, clearly that is a cross-section of a root. That's clearly a dicot root. This is a close-up of the stele. You can notice here the endoderm is highlighted in red. Then you have the pericycle, which is this one cell. Here's the phloem and the xylem, and it has the cross. Now, this is a great picture showing side by side dicot and monocot. So again, here's the cortex, here's the dermal tissue, here's the stele, xylem, phloem, etc. And then over here, dermal tissue, cortex, and then you can see the stele located here. You can see the endoderm is highlighted in red because it has that Casparian strip that stains a little darker. These bigger circles are the xylem. Do you see that? Right there, the bigger circles, and then the pith is on the inside right there, and then here's the phloem. So I was alluding to this before, like if you're going to produce a lateral root, in other words a root that's coming off on the side, you got to bust, it's like a sort of breaking through, you got to bust through the epidermis, you got to emerge through the cortex. And so where does a lateral root originate? Where does it sort of spring from? It springs from this pericycle layer. Those cells become meristematic and they start dividing. Not only do they become meristematic, it's a primary meristem, so it's going to become protoderm, procambium, and ground meristem. So this root is going to have its own ground tissue, its own epidermis, and its own vascular stele right there. And so let's draw our attention now away from the root and look at the shoot. And so if we're talking about primary growth in the shoot or the stem, where is it going to be? It's going to be at the tippity top. So this is I don't know what this looks like to you, like some house right here with the door, but basically this is the top, the apical terminal bud of a shoot. So in other words, these are the cells of the apical meristem that are dividing, mitosis, mitosis, mitosis. And what do they make the three primary meristems same as in the root, thank goodness. So in other words, the protoderm will eventually become the epidermis of the shoot. The ground meristem will eventually become the ground tissue, and the procambium will eventually become the vascular tissue. And so if you take a look at this meristematic region, this apical meristem, you don't need a root cap, but this apical meristem up here on the very top of the plant are the cells that are dividing. And so, as I mentioned before, let me just say it again if you don't mind, just for clarity. The primary division of a plant for growth, primary growth in an upward direction. The reason it's going upward is because the cells are pushing down, so it's going up right here. What you got is protoderm, which is eventually going to become the dermis, procambium, which is eventually going to become the vascular tissue, and ground meristem, which is going to be the ground tissue of the root. So these are your three primary meristems, and they're at the tippity top of the shoot. And so this is a stem. So if you took a plant, an herb, I know it's an herb, it's not a woody because it's not very thick, but if you cut a stem in half, this would be a typical monocot over here, and this would be a typical dicot over here. There's some differences. One of the things that you're looking at is this vascular tissue, and these vascular bundles. Do you notice here, these vascular bundles in a dicot are around the perimeter of the stem. This is, again, pithy area in the center, so they're around the perimeter like that. Whereas look here in the monocot, the vascular bundles are sort of scattered throughout the stem. You see that? Now, you wish you had higher magnification, and so yes, I agree. So here's the dicot. Here's the pith in the center. What's the pith doing? It's providing some strength and some stability along with the cortex. The cortex is not so much a function in the root as storage, but it provides strength. And so the cortex, if you remember this, is made up of parenchyma cells, which have a little bit of a thicker primary cell wall. If you look real close at one vascular bundle, you'll notice how the xylem is more towards the center and the phloem is a little bit more towards the outside or cortex. So you got dermal, ground, vascular, ground right in here. And so this is a dicot. Again, the dicot, and here's the monocot. Notice the vascular bundles are sort of spread out in between. So dicot, monocot. Here's a close-up of the monocot stem. The monocot stem has these little vascular bundles, but what's interesting is botanists, I guess trying to be funny, maybe, I called these vascular bundles little monkey faces because they look like they have eyes and a little bit of a nose, looking like a monkey. I don't know, maybe. Here's the eyes right here, the monocot stem. These are obviously the very large xylem vessels. Notice how thick they are. They carry water. Here's the phloem. I like this picture a lot. Look at the sieve tube members, which are also hollow, but then they have these little companion cells, which are really helping out right there. You know, this doesn't have a large amount of cytoplasm right there. So this is a close-up of a vascular bundle of the stem of a monocot. And then getting to the leaf. You know, the leaf has this upper area right here, which is dermal, and then it has this ground tissue, which is called in the middle, or mesophyll. The vein is not shown in this picture. It has this waxy cuticle. And the epidermis, a single cell layer, it's sort of like a puzzle, is the first line of defense against pathogens, and also the cuticles to prevent water loss. And so what's fascinating, you might be familiar with the leaf from photosynthesis, but if you looked on the underside of a leaf, you'll notice that there's these specialized cells that flank these pores called stoma. These specialized cells are called guard cells, and they're capable of photosynthesis and opening and closing. They swell open, which allows carbon dioxide to come in and oxygen to leave. And so here we have is the dermal tissue, and then the yellow is the ground tissue, and then we have more dermal tissue here. And you're like, where's the vascular tissue? The vascular tissue is right here. We call it a vein. And the vein is made up of, surrounded by bundle sheath, if you remember that, the bundle sheath cells which surround the vein. And then inside we have the xylem, which conducts the water to the leaf, and the phloem, which conducts the sugar that's being produced in photosynthesis, dissolved in water, away from the leaf, down the petiole to the stem, and then ultimately the root. Or it could be to a fruit. And so again, we'll talk later about how guard cells regulate transpirational losses, but they do. They close up at night and they open up in the morning generally. But this is where gas exchange takes place, like these little mouths right here. So gases go in and out, and also water goes in and out. Water leaving is a good thing, transpiration, because it pulls through capillary action, that's cohesion and adhesion, it pulls water up the plant. And that's how water is able to travel up xylem, is through transpiration. And so that's providing the pull, especially if it's windy, especially if it's sunny. You have a very low water potential in the air, so the water is able to leave. What's troublesome though is excessive transpirational losses can be lethal to the plant, and so that's a cause for concern. And so you can see here, dermal, here's ground, and vascular in the leaf. There's the stoma, the guard cells, which allow the gas exchange. Oxygen out, CO2 in. And that's the mesophyll, which is ground tissue. These are parenchyma cells, these are palisade, these are spongy in relationship to the way they're close to each other, like a palisade, like a fence, which captures a lot of sunlight. Spongy allows air circulation to occur in the leaf. It's really a cool structure. This is one of my favorite organs of a plant is the leaf. It's an incredible example of structure related to function. It's awesome. And then finally, the vascular tissue, I was mentioning this before, travels from the petiole into the leaf. Then it goes into, it brings water to the leaf for photosynthesis, because as you know, inside the plant cell, inside the palisade, the chloroplast needs water, especially in the thylakoid membrane in photosynthesis in order to take place. So here you go. Dermal, ground tissue, palisade, spongy, lower epidermis. Notice the lower epidermis has the guard cells. Notice these air spaces allow for circulation of gases. So I hope you enjoyed this discussion on primary growth in plants. Thanks for watching.